Morning, good afternoon, good evening. I hope you guys are doing all right. Welcome to week 3 of Biology 1021 Cell Theory. My name is Ian Coates, and I'll be your host and facilitator for this week and all weeks. Again, I hope you guys are doing all right. Please make sure there that you guys have looked at weeks 1 and weeks 2 before going on. You will see a little bit of repetition there from weeks 1 and 2 because that's just the way biology works here. We learn something one week and then we apply it there to the next. So please make sure you've had a look at weeks 1 and 2. Let's get on with week 3, shall we? You can see what we're looking at here. There's a eukaryotic cell. Look at the different types of cells that are here. There's RBCs, there's a sperm cell, macrophage, there's a nerve cell, muscle cells. We'll have a look at those. And here's your DNA and chromosomes. So we're going to hit all these as well as some membrane transport. Let's go, shall we? Okay, introduction there. The basic organizational structure of the human body is the cell. Okay, we are nothing but saltwater bags, as I said in the last video. All right, and the only way to make little compartments, aka cells, okay, in something that is made of water is to make the cells out of fat. And you'll notice there that the cell membrane of these cells here is made out of fat, not water soluble, so the water inside is actually separated from the water outside, which is the whole point. There are anywhere between 50 to 100 trillion cells in the human body. Quite fantastic. Differentiation is when cells specialize. Nerve cell, muscle cell, bone cell. As a result of this differentiation, cells vary in size, shape, due to their unique function. All right. So, cell theory. Here we're talking about the living units. A cell is the structural and functional unit of life. How well the entire organism functions depends on individual and combined activities of all of its cells. Excuse me. Structure and function are definitely complementary, and it works the same way in the microbial world as well. Okay, remember anatomy, physiology. Anatomy is the structure, physiology is the function. Biochemical functions of cells are dictated by shape of cell and the specific subcellular structures. Now that's some good coffee. Continuity of life has cellular basis. Cells can arise only from other pre-existing cells. And you look at that sentence, it's pretty obvious, but you got to say these things in science in order for them to be actually true. Of course, cells arise from, other pre-existing cells. And basically what we're saying here is, cells just aren't being created out of magic, just kind of arising from nothing. Something has to come from something. Okay. Cell diversity, over 200 different types of human cells. Types differ in size, shape, and components. These differences lead to differences in function. And there, just from the title slide there, specialization of cells. Here we have erythrocytes, also known as RBCs, red blood cells. Here we have epithelial cells, and there's lots of different kinds of epithelial cells. Okay, we'll get more into these there towards the end of the course. Skeletal muscle cells, smooth muscle cell. The third kind of muscle is cardiac. Guess where that one is only found? Fat cells, okay, macrophages, I call these Pac-Man, chomp chomp, okay, these are phagocytic, okay, they chomp other bacteria, foreign invaders. Nerve cell, very obvious and easy to detect because they're of their unique shape and structure. And here is the spermatid, okay, sperm cell. All have different functions. Why? Because they all have different shapes and sizes. The generalized cell, Okay, all cells have common structure and function. Human cells have three basic parts. Okay, there's the plasma membrane. There's your fat. Okay, flexible outer boundary. Cytoplasm, call this there the jelly or the quote unquote blood of a cell, right? Things got to move from one side of a cell to the other. How do they do it? They do it by the cytoplasmic matrix there, this intracellular fluid containing all of the organelles. The nucleus, again for eukaryotic cells, not prokaryotes. That is where the DNA is housed. Okay, just had a nice easy shot. And you know, you can go into any introductory biology textbook and find a similar cartoon. Okay, you just have a look here. We'll just move on. We'll go from the nucleus and around in this direction with no relative importance to anything here. There's your nucleus, okay, within the cell. And you'll see there that this is what we call the nucleolus, your chromatin, your DNA is housed in here. You'll see that there's holes in the nucleus. Okay, that's because things have to go out and come in. We'll talk more about those as we get there. Okay, just the outside here, there's your plasma membrane. Okay, you have the rough endoplasmic reticulum, sometimes called RER. You'll have ribosomes floating around, usually in the rough ER as well. They can be free ribosomes. There's your Golgi apparatus involved in packaging. And you can see some proteins there. They are getting made and packaged and sent outside. Okay, could be hormones as well, depending upon the cell. Now the cell has a shape, and you see this cell here is kind of, you know, circular, you know, sphere-like. And you'll see how is that sphere-ness? How does that shape maintained? You'll see here, zooming in, you'll see these microtubules. We call these there the cytoskeleton. And it is in fact holding that cell open, giving it its shape. Okay. These orange peanuts right here, okay, these are very important. These are called mitochondria. These are the powerhouse of the cell. This is where ATP is made. The whole idea that oxygen goes in and we extract energy from our food particles. There we go. That's the mitochondria right there. Centrioles, lysosomes, okay. There's your cytoplasm, the gel matrix. Here's your smooth endoplasmic reticulum, not to be confused with your rough endoplasmic reticulum. Peroxisomes, okay, these guys here are involved in garbage collection. It happens. We'll get there. Let's go. You remember there, prokaryotic, these are the bacteria. Okay, quite different. Okay, this is much more highly organized. You can see here, this is just kind of a hodgepodge. But not to take away from their effectiveness, these things are very effective. As I said before, they were here billions of years before us, and they'll be here billions of years after. No nucleus, no membrane-bound organelles, just ribosomes. What do ribosomes make? Ribosomes make protein. All prokaryotes are unicellular, forerunner to eukaryotic, okay, smaller, more simpler. A single closed circular chromosome contained within the nucleoid, no histone. So it's just a circular piece of DNA. So there's a starting point will be exactly the same as the end point. That's what makes bacterial DNA very easy to replicate. And that's why the life cycle of a bacteria is only 20 minutes. Even has additional circular DNA. We call this their plasmid. This one here is involved in antibiotic resistance. Okay. And you'll see here there that the cell wall, we talked about this there a little bit there last week there, the whole idea that it has this chemical called peptidoglycan. And this is involved in the creation there of the cell wall of the bacteria, and some antibiotics, they attack this peptidoglycan and therefore creating a hole in the bacteria that the contents can leak out and the bacteria dies. Smaller ribosome units here, it's all about protein synthesis. And bacteria, they divide by binary fission. Okay. and that's where one cell will create two. Okay, and this is not mitosis. They'll also have a flagellum as well for locomotion. Okay, so a bacteria appendage there, it has what we call a sex pillae, and you'll see here in this electron micrograph here, this bacteria here is sending information to this bacteria here, okay, and it's sending those plasmid DNA molecules from one to the other, and that's how quickly this whole idea of antibiotic resistance can spread through a colony. And this can even happen between bacteria of not the same species. Okay, so these pillae are short hair-like proteins, function adherents. They stick to each other, stick to surfaces, harder to wash away. The sex pillae, that's about conjugation. Okay, good. And conjugation, sorry, that's the transference of information from one bacteria to another. So, back to eukaryotes, extracellular material, so substances found outside the cell. Classes of extracellular material include, okay, extracellular fluids, body fluid, okay, blood plasma, CSF, cerebrospinal fluid, okay, and this is fluid there that circumvents the brain as well as the spinal cord there to alleviate. It's kind of like the whole point is when you go swimming, you don't weigh 100 kilograms anymore, you only weigh 50 kilograms, right? The water creates buoyancy. The water does the same thing there with the brain. Cellular secretions, saliva, mucus, extracellular matrix substances that act as glue to hold cells together. Okay, so let's have a look. Inside the cell, here we've got the cytoplasm, and here's the same shot as before. You can blow this up as needed. Okay, I just put this here and just blow it up, right? So there's your cytoplasm, okay, a gel-like substance found inside the cell but outside of the nucleus, okay, composed of cytosol, kind of like a jelly-like matrix there, but it's primarily water, electrolytes, nutrients, and houses the organelles, which are the little organs of the cell. The plasma membrane, as you can see it here, remember those hydrophilic heads and hydrophobic tails, right? Aha. So it's the outer limit of the cell itself, controls what moves in and out. Love this word, selectively permeable, meaning there it's semi-permeable. Some things can get in easy. Smaller molecules, oxygen, carbon dioxide, sugars can pass freely through. But bigger molecules like proteins, no. They have to be carried in, okay, through proteins. Phospholipid bilayer, there's your water-soluble heads, water-insoluble tails, permeable to lipid-soluble substances, okay? So lipids can move freely through. Cholesterol stabilizes the membrane. So you see these glycoproteins here, okay? That's how we tell our cells from somebody else's. So if a foreign cell came in with different glycoproteins there, our immune system would detect different glycoproteins and all of a sudden sound the alarm bells. All right. Just another shot there, okay, of the cell membrane. You'll see there that it's not just a thin piece of plastic. It has proteins embedded into it, glycoproteins there. The whole idea there, selectively permeable. So the cytoplasm, mostly water, it holds there the organelles, is really like a jello fruit salad where the jello is the cytosol and the fruit, oranges, grapes, bananas, maybe some walnuts, are the organelles, and they're kind of held into place by this jello-like substance, the cytosol. The nucleus, the largest of all the organelles, contains the genetic library of blueprints there for synthesis of nearly all cellular protein. And that's one of the big things that we go back to the DNA for, because we can store sugar, we can store fat, but you can't store protein. And we need protein every day. Our hair grows every day, our eyelashes, our eyebrows, our fingernails, toenails. cartilage needs to be re-updated, and you skin your knee, you need proteins there. You have to have proteins on a daily basis, okay? And that's what we go back to the DNA for, is that the cell will need a protein, well then the DNA will open, we make a copy of it, of that DNA, and make a flimsy copy RNA, and from that there we make proteins, which we'll get into sooner than later. Responds to signals that dictate the kind and amount of protein that needs to be synthesized. Absolutely. Most cells are uninucleate, one nucleus, but skeletal muscles, certain bone cells, and some liver cells are multinucleated. They have many nuclei within a particular cell. Red blood cells are anucleate, no nucleus. Red blood cells, there's no mitosis, they don't divide, but after about 4 months they get the chomp and they get broken down and new RBCs are made. The nucleus has 3 main structures: the nuclear envelope, nucleoli, and chromatin. So here's your nuclear envelope, okay, and you'll have a look here. The outside, there's your nuclear envelope. Porous double membrane separates the nucleoplasm from the cytoplasm. So it creates an in and an out. Remember cytoplasm, and inside here is what we call nucleoplasm. Just the same thing, but inside the nucleus. Nucleolus, okay, right in here. Dense collection of RNA and proteins, site of ribosomal production, okay. And then you have your chromatin, fibers of DNA and proteins, stores information for synthesis of proteins. Tightly coiled chromatin is what we call a chromosome, okay, though it kind of looks like tightly wound yarn. So, looking at the nucleus here, controls the working of the entire cell, is surrounded by a double-layered nuclear membrane, okay, that's your envelope, okay. Found inside the nucleus there's nucleoplasm, just a gel-like substance similar to cytoplasm, the nucleolus, okay, involved in the synthesis of ribosomes, and then you have thread-like structures there that contain the genes. That is your chromatin, and that's found all in here. Good. The nuclear envelope is a double-membered barrier that encloses the jelly-like fluid of the nucleoplasm. Outer layer is continuous with rough endoplasmic reticulum, and like the rough ER, is studded with ribosomes. So rough ER, rough endoplasmic reticulum, not smooth. That's a different ball game. That's a different way, even though they kind of have the same name, endoplasmic reticulum. Rough ER has ribosomes studded with it, and ribosomes make protein. The inner layer of the nucleoplasm there, called the nuclear lamina, is the network mesh of proteins that maintains nuclear shape and acts as the scaffolding for DNA. Okay, holds the DNA together. That's your nuclear lamina. Nuclear pores allow substances to pass into and out of the nucleus. They are guarded by the nuclear pore complex, which regulates transport of specific large molecules. Okay? Very good. The DNA, the chromatin itself, okay, consists of 30% thread-like strands of DNA, 60% histone proteins. These are these little, you can see them here if I zoom in. You see these little round discs right here? Those are what we call histones, and that's what the DNA is wrapped around. And you'll see how many histones there are. There's bazillions of them, right? That the DNA is all wrapped around and protected. Okay? So 30% DNA, 60% histones, and 10% RNA. Arranged in fundamental units called nucleosomes, which consist of DNA wrapped around the histone. Okay, that's called a nucleosome. Chemical alterations of histones have an effect on DNA and therefore can help regulate gene expression. Chromosomes are considered chromatin. Condensed state helps protect fragile chromatin threads during cell division. Okay? Good. Cytoplasmic organelles, these are the ones there we're going to hit. These are the main ones there. You've got mitochondria, powerhouse, ribosomes, protein, endoplasmic reticulum, rough and smooth. We've already talked about rough. Golgi apparatus for packaging, lysozymes, okay, cytoskeleton, centriole, microvilli, cilia, and flagellum. Let's go. So Golgi apparatus, this one here is for packaging proteins. That's what's going on here, okay? And the whole idea, proteins cannot get through the membrane. And if a protein has got to go, okay, then it has to be packaged. It really does. And that's what's going on there. I'm just changing the color on that one. Good. And you'll see protein is made, and then it's packaged up, and you'll see there that this will eventually make its way to the membrane, where this will become part of the membrane, and the protein or hormone or whatever is being made gets released out. Okay? Mitochondria, okay, this is the powerhouse of the cell. There's two layers involved. There's the smooth outer layer and the inner layer folds called cristae. These guys in here, okay? And the inner layer contains enzymes involved in ATP production. And ATP, as you know, is our energy currency. If we need to move our muscle, we spend ATP molecules, which we get from eating food. The mitochondria are tiny slipper-shaped organelles that are the power plants of the cell. They produce most of the energy, ATP, in our body. The cristae contain the enzymes associated with ATP production, and that's these guys here, the cristae, these inner structures. The number of mitochondria per cell varies because of the metabolic activity of that particular cell. The harder a cell works, the greater the number of mitochondria. Okay. Just another shot here. You have a cartoon next to a real electron micrograph of mitochondria, and you'll see these cristae, these inner folds there in the middle. Okay. We are in good shape there. All right. Enzymes there on the inside. Okay. ATP is a molecule that stores energy. Energy is stored in the bonds that attach the phosphate to the adenosine part of the molecule. Okay, and here, that's how we extract energy from gasoline. If you look at octane, it's just a hydrocarbon. It has hydrogen and carbons. That's it. And the whole idea of burning that, we break those bonds between the carbon and the hydrogen, and when we break those bonds, energy is released, and we're able to capture that energy and make our car go. Our bodies are able to capture that energy and store it as ATP. Okay, so ATP here, this is the one that has energy. We utilize it, and the energy is released. We're able to lift that barbell or run for a kilometer. And what are we left with? ADP, adenosine diphosphate. We eat another banana, we make more ATP, and this is how the whole system goes. Okay. Ribosomes, they are found within the endoplasmic reticulum. They're found free. They're also found in the nucleus as well. These guys here, they have one job, okay, and I kind of look at it as like a Big Mac. There's a top unit and a bottom unit here. And what happens there, that copy of the DNA, an RNA, okay, copy leaves the nucleus and travels through here like a stock market ticker tape. And this guy here reads that RNA template and makes proteins and takes an amino acid, another amino acid, another amino acid, and eventually makes a big long protein chain. Okay? Provides structural support for RNA molecules that come together for protein synthesis. Endoplasmic reticulum, network of membranes in the cytosol. There's the rough ER. This has fixed ribosomes on the surface. Okay, and you'll see here, it's not just membranes here. You zoom in there, you can see little ribosomal complexes all studded in here. Smooth ER, though, there are no ribosomes. This is the site of lipid and steroid synthesis. So rough ER is involved in protein synthesis. Smooth ER is involved in fat and steroid synthesis. Got it? Good. Waste management, lysosomes, peroxisomes, centrosomes here. Here we're talking about these guys here have enzyme-containing sacs that break down waste. Okay, that's what's going on here, the peroxisome as well. While the cell is perfect in a lot of ways, there are little bits and bobs of protein there that get left behind, and that's what lysosomes and peroxisomes do. Centrosomes, these are two rod-like centrioles there used to produce cilia and flagella. Okay, very specific proteins there, distributes chromosomes during cell division. Okay, good. Cytoskeleton, these are those microtubules, okay, microfilaments there that are hard to see unless you stain them there, and you can see, and they hold the shape of the cell. Okay, kind of like the skeletal structure of the cell. Microvilli, these are found there in the small intestine. The body is an amazing place for creating space where there isn't any, and the whole idea there that with these microvilli, you're able there to increase the surface area of the inside of the intestine. You know, like a small little area will now have the surface area of a tennis court, which is why we're able to digest and absorb all of the nutrients from our Big Mac combo. Okay, these are called microvilli, increasing surface area. That's what they do. Cilia, these are short hair-like projections there found in the lungs. Okay, and you kind of think of them as seaweed at the bottom of the ocean, that they kind of move with the current. And that's the whole point there, is that you'll see there with the mucus layer, the cilia moves this mucus that will definitely have bacteria or smoking particles or some toxic chemicals lodged in there, and that mucus gets moved out and pushed up the trachea to eventually to a point where it can be spit out. Okay, flagella. Here's another aspect of a cell here, very highly specialized. When you're talking a sperm cell, you'll see there this protein tail. It's able there for locomotion and movement, enables the sperm to swim. Okay. Here's some general characteristics there between prokaryotic cells and eukaryotic cells. We've covered these, okay, as well. Like when you're talking prokaryotic, it's strictly asexual reproduction, but eukaryotic, there is the possibility of both. Bacteria divide by binary fission. Eukaryotes there through meiosis, reproduction. And here's some more. Okay, here we're talking about the lipid bilayer associated there with the membrane. Okay, the cell wall, basic structures differentiating there between the prokaryotic and eukaryotic. Here we have some differences of the functions that go on between the two. Okay, remember there that your eukaryotic cell is more highly advanced, okay, than your much earlier prokaryotic. Okay, have a look at those. Good stuff there. More functions. You know, as far as different types of ribosomal complexes, I'm not too interested. Okay, of course, bacteria are going to have different kinds of ribosomes associated with them, but 70S and 80S, I'm not too worried about that. Okay. Movement in and out of the cell. This is why we have a cell membrane, okay, because some things have to go to this cell, some things have to go to this cell. There is this differentiation, and you know, we're we're selective creatures, and we're specialized, and so are our cells. So there are two ways substances move across or through the cell membrane. There is what we call passive, and there is active. Okay, passive is free money, no ATP involved, and you'll see there the whole idea. The body utilizes this as much as possible because it's free money. the whole idea of breathing. There's a greater concentration of oxygen in the air than there is in your blood. So when you take a breath, the oxygen goes into your lungs, into your alveoli, and immediately jumps into your blood because it has a greater concentration on one side, and it'll always flow to a lesser concentration. So simple diffusion, free money. Facilitated diffusion, again, moving from a high concentration to a low. Osmosis does the same thing. That's the movement of water. Okay? Filtration. There's another one. All of these four are free money. Doesn't cost the body anything. Now, but if we want to move things from a low concentration to a high concentration, this is where we're going to need money. And when I mean money, I mean ATP. Okay? And active transport, endocytosis, exocytosis, transcytosis, all of these processes here, they cost money, ATP. We're going to talk about these right now. So, simple passive diffusion, the process by which molecules or ions scatter or spread spontaneously from a high concentration to a low concentration. Atoms can diffuse readily, even through the cell membrane, until equilibrium is reached. Okay, so the whole idea here, you'll see what goes on. I have a higher concentration here, and I have a high concentration of blue here. So if the particles are allowed to move, then you'll get a nice equilibrium eventually over time. A vitamin C effervescent tablet, right? Over time, that effervescent tablet will completely break down, okay, and form an equilibrium. A skunk, I don't know if you've ever been hit by a skunk at ground zero. I have. A skunk smells very different at ground zero as opposed to being a kilometer away, 3 hours after the fact. So it will passively diffuse and form an equilibrium with the entire environment. Free money, simple diffusion, love it. Facilitated diffusion. This is where a helper molecule within the membrane assists with the movement of substances from a high to a low concentration. So, some things there can just move freely across a membrane. Some things cannot, and they need a carrier protein. And that's okay. If you have a high concentration of one side, this will allow it, and these particles will be allowed to diffuse through. Facilitated diffusion by using a carrier protein. Osmosis, we talked about this there last week. Here we're not talking about the movement of particles like we were talking about with diffusion. Okay, here we're talking about the movement of water. And water will move from its high concentration to a lower concentration of water. Okay, and you'll see there that high concentration of water to a lower concentration of water. This water here, because these particles can't move for some reason, this is what we call a selectively permeable membrane. Particles can't get through, but the water can. The water will move from its own high concentration, and you'll see there that now we have an equilibrium. Okay, we saw this slide there last week. I love it. Isotonic, when you take an RBC and place it into the same salt concentration as it's used to, you get it looking nice and nice, where the salt coming in is equal to the salt coming out. Okay, if you place an RBC into a salt water concentration that is higher than what it was used to, the water will leave the RBC and travel into the salty water. That's what we call a hypertonic, a greater salty. That's hypertonic, and hypotonic, less salty. So therefore now the water rushes inside the RBC and it explodes. Okay. Filtration. This is a great one too. The whole idea there that if you increase the pressure, okay, it relieves it on one end. It's called Boyle's law, right? If you increase the pressure, you're decreasing the volume, comes out. If we pull on the plunger, okay, you're decreasing the pressure, you're increasing, okay, the volume inside. Okay, so filtration is another good one as far as free money go. And you see the body, it utilizes it all the time. The pressure there from the heart forces the RBCs out into the cells. Okay, so the movement of water and dissolved substances from an area of high pressure to an area of lower pressure. Water and dissolved substances are pushed. Okay, good. Just another shot of the same thing. Okay, smaller molecules are forced through porous membranes. Hydrostatic pressure is important in the body. Molecules leaving the blood capillaries. They're filtered out. They're pushed out into the tissue fluid. Okay. Now those are the 4 there that we talked about that were free money. Now we'll move on into the ones there that cost money. Here we're moving from a low concentration to a high concentration. We're kind of going against the laws of nature here, okay? Kind of like pushing the water up the waterfall, right? So this one here is going to cost us money, and we call this active transport pumps. So moving substances from an area of low concentration to an area of high concentration requires ATP, and equilibrium can never be reached. Okay, there's no kind of diffusion where everything is the same because you're going against, you're pushing that water up the hill. So you've got endocytosis, okay, and you'll see there, just remember your Golgi apparatus there, they're packaging up a protein, and that protein's got to get outside the cell because the cell makes this protein. So it packages up the protein and travels out, okay, and is released, okay, but then you can have the opposite, okay. That one there is called exocytosis, moving in this direction, and then you'll see endocytosis, and this is the whole idea of bringing something into the cell. See, there's your exocyte. It's exactly the same process, you're just changing direction. So I'm just trying to say the Golgi apparatus, they package things up. So going back in here, endocytosis, ingestion of substances by the cell membrane. There is phagocytosis, chomp chomp, Pac-Man, where one cell is going to digest another. Penocytosis, this is if a cell is going to ingest something that's mostly water. So phagocytosis, you're ingesting a solid. Penocytosis, you're ingesting mostly water. And then you can have what's called receptor-mediated endocytosis. You see here, maybe there's a hormone floating around or something, a molecule floating around, and there are particular receptor proteins on the outside of a cell. These things latch on, and then the cell knows to bring these in, and that's what goes on here. And now those guys are inside the cell. Okay, so this is endocytosis, bringing something in, and you see the membrane there invaginates, pinches off, and now it's able to bring this into the cell and utilize those particles as needed. Endocytosis, bringing in. Exocytosis, bringing out. Okay, the whole idea there, Golgi apparatus packages something out and sends it on its way, and then you'll see there that this eventually just becomes the new edge there of the cell membrane. Okay, release of neurotransmitters from nerve cells, all sorts of things there that we use this for. Transcytosis, good example here. The whole idea here, we're talking about HIV, crossing a cell layer. HIV, as you'll know there, it goes after white blood cells, okay, and that's where it develops and matures, okay, and if it sees a white blood cell on the other side, it has the ability there to go endocytosis and then exocytosis. Both of these in the same motion are called transcytosis. Okay, transport a substance rapidly through a cell, and that's what makes HIV so dangerous. Okay, and I think that takes us to the end of our PowerPoint. You'll see there that we have all some good questions there. Let me know if you guys have any issues with any of these. Okay, and I think you guys are wonderful. Just keep at it. Please make sure that you've read the readings there for this week. Next week there, we're on to Microscope Lab. So, talk to you guys later and have a wonderful and safe week. Take care. Bye-bye.